Thank you. Thank you, the organizer, to having the trust and inviting me to present. I am honored to be here. I am very new to the field, and I am looking for your feedback on my presentation. I am the head of the preclinical core at Regeneron, and to give you an idea, we are handling all possible species. We are working on mice, rats, non-human primates, zebrafish, rabbits, you name this. Humanized mice, any kind of model but human. And our most asked service is the validation of the probe for the antibodies and for the RNA probes. So we had to come up with an idea, almost like SOP procedure, how we handle this massive flow of different target tissues, species, and how we address this need in a careful but yet efficient antibody validation. And that's what I would like to show you today. On a high level, the antibody validation should address the question, what is the truth? And in this case is, what is the true pattern? You are asked to have the antibody for F480 in liver, and you buy one commercial antibody, you buy another one, and they produce quite different pattern. Which pattern is the true one? Let's step back and dissect a little bit this question and to the bite size. What we are trying to address is where is here the signal, what is the background, and what is the off-target. And that's what I want to show you how we go through this question in a quite efficient way. So we do rely on the 5 pillars of the antibody validation, and we also have some other experiments that we use for the validation of the targets. We are privileged to work in the pharmaceutical company, and sometimes we have different controls, which is typically not available in other spaces. I will not stay long for the antibody pillars. I will show you on the next slide how we put them in a way it makes a standard operation procedure for the antibody validation. Here is the flow. I will just show you how we do this, the frame, and on the next slide I will show you an example of how we apply that. First, we start with the antibody selection, and we typically select 3-4 antibody against different epitopes. We like the paired approach, and 2 historic or well-known antibodies if those are available. Second, controls are no less important as the antibody themselves. So we use RNA databases. We have internal database, but also public database to identify high, medium, and low expressor. We want to see that our antibody works on the high range of the endogenous proteins. And then, since we are in the preclinical research, we look what other controls are available for us, which is coming for the requesters. Do we have knockout mice, reporter mice, stacked protein, genetic treatment? All of this in application of the tissue. This is not the cell lines. Then we also have the choice of non-genetic treatment, cell lines, which could be knockout, or it could be the cells high or low in the expression. And in some cases, we have no special requests. Depending on the confidence in the samples, we use, in general, 2 workflow. One is for the very confident examples, like a knockout, reporter, target protein, basically when you have black and white, like yes or no response. In this case, we would have a multi-tissue block selected from the RNA-seq databases. We combine them and run the antibodies on the positive and negative animals, providing that those animals are confirmed and you do know from orthogonal assessment that this is the right knockout or knock-in. And then you look for the consistent pattern. Basically, the positive animal should have the positive pattern and negative should have negative. It gets a little bit more tricky as we go for the less reliable controls, which also, in our opinion, include genetic treatment, since this is not a full knockout or full knock-in. So we get a little bit more cautious with this. So we do apply additional validation sets. And in this case, it includes orthogonal. This is RNA scope or molecular instrument to identify RNA and see how it correlates with the at least location of the protein. And the second, we do use a paired antibody. Paired antibodies are especially important if you go to the brain and you have the cells with a long processes and you want to be sure that the processes that you see are indeed positive for this target. So once you identify what is the pattern, you actually can go to optimize this pattern, which is basically optimization of signal to noise. And that is usually achieved by the dilution of the primary antibodies. Here is an example how we do this. And I selected a very simple markers just to demonstrate the approach. This is the F480 markers that the requester wanted to use in the liver. So the pillars that we are going to use are paired antibody and two orthogonal assessment, RNA-seq and RNA-scope. And we don't have any kind of special controls. So first, the antibody search. We start typically with the site AB. This is our preferred platform to look for the antibodies. And if the target is as known as F480, you can be very picky about what you put here. You could have primary rabbit, our detection is tailored to the rabbit antibodies, immunofluorescence, IHC paraffin, and this is in the mouse. In reality, many targets that we have are so new that I eliminate everything but the primary and maybe the origin where it's supposed to be. So you could see that in this particular antibody, 91 antibody came up, and the leading one is the CST antibody. So we will use this antibody in the test. This was one approach. The other approach is, especially for the targets which are less established, just goes to the manufacturers that we initially have a very good history of performance of their antibodies. I just show one of them, so this is by far not the only one that we work with. This is a very good way to demonstrate that they have a full description of the protein. And what is very important for us, they show at least approximately where is the epitope of antibodies. We do rely on the paired antibody a lot, and we do appreciate when the vendors put this information. That is very helpful for us. What comes also here, you see that the antibodies are validated, and if you press this, you will have the menu. This is the assays which will perform. One would hope if you press a button and click on the independent antibody, you would have the pictures with the two independent antibody producing the same pattern. But instead, this is just a general description. So we wish we would have more original data confirming these statements. What looks promising here is the FFPE section of the liver with some of the cells. And if you know how the F480 should look like, that already looks like promising in the sense of Kupffer cells. But for this, you need to be an expert. We would like to be a kind of unbiased, and that should not require expertise to figure out the antibodies. So finally, we selected several antibodies as I promised, and this is just an example of what we have. We have upcome section signaling that antibody which came from the site AB. What is important for us, we need to know the host, the reactivity, and we do prefer again to see the epitope or immunogen which would use to create the paired antibodies. All antibody we run on like a bond platform in an automatic way, and we use high polymer detection for all of the antibodies. So essentially, they all run on the same protocol, same retrieval. We do use two platforms for the RNA detection. This is ACD and molecular instrument. Molecular instrument allows you to combine the protein with the RNA probe in the same section, as it doesn't use the protease. So next, control selection. So we use internal Regeneron database for this. And what we are looking for a wide range of the expression. We want to have low expression, pancreas and cerebellum. We want to have medium expressor, which is highlighted here, skin and liver. And then we are looking for the high expressor, like the group of the lymphoid tissue which stand out. The lymph node seems to be great, but instead we are taking spleen, because spleen is the large organ, and we would have multiple validation using this organ as a representative of the lymphoid tissue. So when it's selected, we literally put this in a block. So we have a luxury because the mouse is small, literally combine the large tissue and have all this diversity and complexity of for each tissue on one slide. We could typically combine up to 10 tissues, and we have variety of the multi-tissue blocks, which include major organs like this, but we can get also very nuanced when we have eyes, glands, and all the other parts of the mouse for extended validation. And this is the section from the same block, and you already see that the high expressor liver is glowing with this F480 IHC. So how we approach the validation itself, and I will show you literally all raw data. So first we start with the IHC in this case, and you would see this is the liver, and this is the cells, which is a kind of in between the hepatocytes. And then we do need to have a negative control for our assay and the positive control for the RNA preservation in the tissue. And then what you do, you actually compare the IHC with your RNA scope performance. And typically, we test the antibody at 4 concentration. Most of the antibody with our tissue processing and our fixation and detection comes in this range. That's what you do for antibody number 1, APCAM, protein tech, BTIL, APCAM. Again, this is just a few representative. And you're looking for something which is very close to the RNA and close in between the antibody. This is as close as it gets for each of the antibodies. And you see they all have in common this pattern, like the cells in between hepatocytes, Kupffer cells. This antibody has some off-target. This antibody, it's correct, but it has pretty low signal-to-noise ratio. Then you basically apply the same approach for all antibody and all tissue what you have in a multi-tissue block. So you rate each of the tissue for each antibody for strong signal, low background, and no off-target. You want to have all three green. Strong signal and background are kind of coupled, but that's how we rate this. So the Epcam antibody, that one is not so good for the signal to noise. This one has off-target, this one is great, and so on and so forth. The good news is that only one antibody did not perform, like this is the one. What is getting more interesting, let's go to look other tissues. So spleen, the high expressor. The antibody which produce off-target here has perfectly fine signal in the spleen. So this antibody is consistently well on this tissue, and this antibody consistently didn't perform well. So the antibody is kind of tissue dependent, especially the antibody which are not completely optimal. So going even further, this antibody which performed great in spleen did not perform great in the intestine. And you could basically analyze all the antibody. But the good news is there are some antibodies which are consistently well performing, and this is the strong antibody. So 3 companies here came up very nicely, all identical pattern and all strong performer and the right signal, like in lamina propria, for example, in the intestine. Now, what do we do for the report? How we show the data, and how we hope that the vendors and people who publish this in a paper will present this. So these reports comes with every validations we provide for the scientists. We have antibody, we have concentration, we do have methods how we achieve this. And then this is the 2 antibody producing identical pattern. This is our control for the detection, so this is correct, and this is the RNA scope orthogonal assessment. And I also like to add the tissue where people, which is known to express this is spleen. And you do like this for all the tissues that you evaluated in the blog. So I wish people publish something like this, and then you don't need to be an antibody expert or a pathologist to appreciate the level of the validation, and you would really know what to do next, whether this antibody is validated or not. In fact, the antibodies that I showed to you were validated by high school teachers. This is the educational programs that Regeneron provide. High school teachers would come to us and spend 2 weeks with us, and this is one of the projects that they like to do. They like to validate the antibody. They take their experience and their report and go to the students and show them how to validate the antibody and how pharmaceutical company is working. For us, we have this table. This is all incorporated in our LIMS system, where we would rate all the antibodies. The great thing, we would like to have all 3 antibodies on the record. So if one of them is discontinued, we have another one. And if the new tissue is coming, we do test all our leading candidates here. Great. This was one of the easiest cases. Now let me show you something more interesting. Validation of the antibody to the mouse IgE. What do we have here? So we are lucky to have scientists to help us to understand the antibody behavior. IgE is immunoglobulin which is produced by beta cells. It's secreted, but it also binds to many other cells by a variety of the receptors. Moreover, IgE is produced typically at the low level. So the typical approach would not work so well, but we have a non-genetic treatment, which is a known model to reduce allergy. This is the house dust mite exposure. We have a naive mouse and we have the treated mouse. So we decided to go with the paired antibodies. Great. We have a good antibodies and they produce the same pattern. And this is 3 different. And you see that this is typically much higher in the house dust mite treated, the positive control, and lower in the naive. But what is actually the background here? So where do I cut the line? And to represent it here, if you have a very concentrated antibody, any of them would have the same pattern. You would have cell nuclei labeled. You would have a huge, massive areas around some of the cells, very positive, and you also have a background activities. So remember, the IgE is secreted protein. Is this the right pattern? Is this the right pattern? Or do I need to go that low? So I cannot quite cut the line and understand what is the actual pattern for these antibodies. So it's still not clear what is the right signal for this model. Even the paired antibody and the treated model does not help me so much. So being the core, we are a kind of natural hub of all requests coming from us. And I know that I have a mouse from completely different requester which doesn't have a B cell. It's not typically the IgE knockout, but without beta cells, there is no production of IgE. So we could use this instead of the knockout for the IgE. So we are adding genetic knockout pillar to our validation. And here is the outcome. That is what I showed before. And when you add this no B cell mouse, you would see that in the first concentration, this antibody produces non-specific binding to the cells and to the cell nuclei. Here, there is still some background, but this concentration seems to be optimal and great for all the controls. So this is completely clear in the no B cell mouse, and it has some level of activation in the normal naive condition, and this is glowing for the allergy model. So, and that's what I would put in the report for this IgE. So in summary, at Regeneron, we try to leverage the pillars of antibody validation, but we also use some other controls. We are very opportunistic in what we can use to validate the antibody. So it is unbiased and does not rely on a known pattern. You don't need to be the expert in pathology or antibody to appreciate if the antibody validation level is sufficient for your goals. So validation is not one step. It's usually graded and tailored to your specific needs and also resources. And validation is also based, in our hands, is based on multiple experiments, and this is based on cross-comparison between tissue and with different levels of the endogenous proteins and RNA, and we also use a lot of the paired antibody patterns. Antibodies are validated for multiple tissue in a single round, and whatever we use for the validation, this block is also running with all our actual controls. So once somebody orders F480, we will run our positive and negative tissue and make sure that the pattern is identical to what we have validated. This is for the reproducibility reasons. And then each tissue may have a different antibody. You see, for the active antibody, especially for the suboptimal antibodies, the antibody may see this in a high expressor, but may start losing some signal in the medium or low expressor. And that's basically it. Thank you so much, and questions.